I'm Thomas Walker. I'm an application scientist with Agilent Technologies. Melissa Chandler, who's one of the account managers for Agilent up in New York, invited me to give a talk on the 6560. So yeah, I'll be presenting on kind of I'll start with the instruments. There we go. I'll start with the instrument, and then after that we can get into kind of like different operating modes of the instrument, and then kind of how those could be applied. Kind of the three main ones that I picked out are what Agilent calls demultiplexing, workflow, the mobility aligned fragmentation, which several types of ion mobility are doing this, but kind of like Agilent's flavor of the mobility aligned fragmentation using ion mobility. And then the collision-induced unfolding, which originated from Brandon Rutlow's group in Michigan. But yeah, I guess I'll go ahead and start with the instrument. So for those who aren't familiar with Agilent's ion mobility system, it's called the 6560. It's a drift tube, full DC. There's no RF confinement in the drift tube. So essentially, it's ion mobility on the front and a QTOF on the back. Essentially, this is a 78 centimeter drift tube. Like I said, just a DC gradient across the drift tube, and then it recollimates in the back to go through the QTOF portion. Typically, we're in the order of 50 resolving power resolution on for the ion mobility dimension. We do have some ways that we can bump that up for certain applications, but typically if you're just running it in the normal ion mobility mode, you get about 50 resolving power for your ion mobility dimension. These are some of the things that we have done on the instrument. One thing that we, in terms of this instrument and all, I guess, uniform field DC instruments, is that you could do first principles based CCS calculations. So because we know essentially the variables in the Mason-Champ equation, we can solve for the time outside of the drift tube, meaning that we can get direct CCS calculations without having to use a calibrant. So there's kind of two ways you can convert to CCS, whether using calibrants or using kind of a first principles approach. This instrument allows for you to use either. So we can do either first principles or do a calibration and correct for it that way. And so I'll be talking about kind of different modes that we can run the instrument in. Kind of the three main modes that I'll highlight here is MS1. So this is essentially just, we're just passing all the ions through. In other words, we call this QTOF mode if like you're sitting in front of the instrument and running it. Then we have the ion mobility mode, which is essentially a trap and release. So we can trap for 20 milliseconds. Typically, we can change this time, but we can trap ions in the trapping funnel after they come through the high pressure funnel, make it to the trapping funnel, and then we could pulse them into the drift tube for the ion mobility separation. We can operate in a different mode, which increases the dynamic range and sensitivity, because we kind of look at the duty cycle. We're trapping in this regime, we're trapping for essentially 2/3 of the time. So 20 milliseconds over 60 milliseconds. In what we call our multiplex mode, which I'll get a little bit more into, we essentially multiplex the ions. Instead of doing one trap and release event, we're doing multiple small packets of ions. You get theoretically 50% duty cycle because we're trapping 50% of the ions. But that makes the data a little more complicated. We do have to use some algorithms to deconvolute the data on the back end. And then I'll mention here the all ions ion mobility that will be coming a little bit later, but essentially we could do alternating frames of ion load fragmentation. Then we can look at, precursors and product ions essentially, because anything, if we look at back at the instrument over here, the collision cell is after the drift tube. So any ion mobility separation has already occurred. And so any fragmentation in the collision cell does not dramatically, it does not affect the drift time. So we get fragments for that precursor aligned in the drift time dimension. Here's kind of another schematic, another way to kind of look at these. So in what we call QTOF mode, essentially the ions pass through. Theoretically, we have 100% duty cycle, but we're not doing any ion mobility separation. So we're not making an ion mobility measurement. In IM mode, like I mentioned, we're trapping and we're pulsing in the ions, and then we're separating the ions based off size and charge using ion mobility. In this case, for most Agilent instruments, we're using a nitrogen gas, but you can plumb in helium or argon or any other gas that you'd want to use. But generally speaking, we're going to be talking about nitrogen. What can happen, as with any trap, in this case, is well, in any trap, you can space charge, and so we are limited depending on the amount of ions that you're trying to cram in this trap. You're limited by the amount of charges. So typically the trap times are 20 milliseconds, but if you have really high abundant ions and you're filling the trap relatively quickly, you can start to see suppression or loss of low abundant ions from the trap. Another way to kind of get around this is kind of multiplex mode, because we're limited to kind of short packets of ions. So we hardly ever overfill the trap using the multiplex mode. But as you can see here, now we kind of have these overlapping distributions of ions, because now we're pulsing multiple packets of ions at any given time. So in this case, we are trapping 20 seconds and pulsing in. Using this regime, we're typically around the 3 to 4 millisecond trap time, and then we pulse in, and usually you can adjust this, but usually we observe the ions for 60 milliseconds before kind of beginning that next ion mobility frame. So as I mentioned before, I mean, the ion mobility, we're separating based on size and charge, but kind of like liquid chromatography, we can get broad diffusion, which increases our peak width. In this case, we get about 50 resolving power. Now, in this case, we can use this a little bit to our advantage when we go to the high resolution demultiplexing, because we can do small packets of ions that don't diffuse nearly as much, instead of kind of like large packets of ions. And so this is what the demultiplexing data kind of looks like. So we have drift time on the y-axis, m over z on the x-axis, and then we have our intensity is color coded, and then this is the LC peak that's being extracted to give us this 2D, 3D plot here. And so this is the result of the essentially the multiplexing. So we get packets of ions. These are not isomers that we're separating. These are actually packets of ions that are being sent into the ion mobility and then being measured by the mass spectrometer. One thing that we could do, because this is a pseudo-random sequence, we can use a Hadamard transform to then convert that back to a single drift time, and then using a demultiplexing algorithm, we're using the Hadamard transform to demultiplex, but then we can also apply a expectation maximization deconvolution algorithm to help increase the resolution if we have some sense of having two peaks being resolved. So in this case, when you fully run the workflow, not only do we get the benefit of having all of these individual signals coalescing into a single drift time, boosting our signal to noise, but we can also apply that demultiplexing, high resolution demultiplexing algorithm to help increase the resolution sum. And here's kind of a real example of 4 structural isomers of leucine. And so when we run our traditional ion mobility experiment, you can see when we extract these out, they kind of all are on top of each other. And then when we take all 4 isomers and average out the total signal, this is kind of the peak we get. Using the high resolution demultiplexing, we can actually increase the resolution somewhat and get better resolving power for these peaks. So we can start to separate all these forms of all these structural isomers of leucine. So that's kind of the benefit of having the high resolution demultiplexing. It works well in certain circumstances. There is one thing that you do have to be careful for, is that with any algorithm you can over process the data. So that's one thing that you will have to be careful using this technique. But especially if you have any LC separation at all, this tool becomes pretty powerful, as you can see in this case here. And then this is the low and high fragmentation. So we're looking again at the iMobility browser, the Mass Hunter iMobility browser from Agilent. In this case, what we're doing is we're operating that collision cell. We essentially take a measurement with low energy in the collision cell. So we have a low voltage collision energy. Typically, our low is 0. So this is kind of what we'd want to call the parent scan, and we could call it that. So essentially, we're highlighting this peak right here, this 1002 m/z peak. Then you can see this is where it falls in the drift time. So once again, drift time versus m/z. And then when we transition to the high fragmentation frame, we now see fragments correlated to this peak, once again, 1002, that now populate. And because we've already separated based off in the iMobility dimension, any fragments that occur in the collision cell will have the same drift time encoded into them. So if you could get separation on potentially isomers, or depending on what you're running, if you're running peptides and you have isomeric peptides and you get separation, you might see different fragmentation patterns. In this case, I believe this was a polymer that was separated, and so you can see kind of these repeating patterns from probably the polymeric units. So this technique can be pretty powerful, especially if you can make sense of or start to separate these from the other compounds in the spectrum, either by LC or by the iMobility. And kind of the last technique that I'll go over here is the collision-induced unfolding. If you're not familiar with this, it's largely applied to proteins and large biomolecules. Essentially, what we can do with the lens here, and so if you follow Agilent's nomenclature, 6560C is the third iteration of the iMobility, and this lens was added. Essentially, gives us a very fine control over the ions exiting the ion transfer capillary from the LC source, or from, if you're doing direct infusion, your ion source here. That's where this lens sits, and especially in this high pressure region, we can now control the energy of ions accelerating into the ion funnel. The thing with the way we operate the collision-induced unfolding experiment is we step up this voltage, so we accelerate our ions as a function of time, and so we kind of follow the stepwise pattern, increasing the energy, and we can kind of get these results here, and I'll explain this a little bit more in the next slide. But in this case, you could take a protein or anything with the structure necessarily, but usually we're using this with proteins. And as we boost the collision voltage, we can start to unfold the proteins in the gas phase. And as you unfold something, you can think it's getting more extended. As it tumbles through your iMobility drift tube, it's now colliding with more of the background gas. That's where that separation based off size and charge comes in. So now as we unfold this protein, it now has a larger cross section. It's now hitting more of the background, more of the nitrogen in the drift tube, and we should see an increase in drift time as a result. So it's getting held back more by all the collisions. And then when we plot this as a function of drift time versus collision voltage, we can then kind of start to see, unfolding patterns, albeit gas phase unfolding patterns, but we can use this as a good comparison tool. It's kind of what I'll have in my last slide of the presentation here is kind of comparing stressed and unstressed antibodies, antibody subunits in that case. But yeah, once again, CIU, increasing the collision voltage, should start to see some unfolding patterns, and we can plot those unfolding patterns into a CIU fingerprint. So we took this and we kind of combined it with another strategy Agilent's been using on the other high resolution systems. This is what we like to call microdroplet reactions. And in this case, just to briefly explain what we're doing here, is we're in this specific data set, we had an antibody, it was NISMAB, so 148,000 Daltons. And typically with antibodies, you have heavy and light chains. And one of the reactions that we can do is a reduction. In this case, I took DTT, which can reduce the disulfide bonds that are present in most antibodies. And in this case, we were able to separate essentially do this reaction by mixing the reducing reagent and the antibody at the sampler. So we have an automated sampler method that it will grab from 2 vials. It will mix from the vial and it will inject it. In this case, we're directly injecting it onto the mass spec. So a reaction that, depending on your temperature, usually takes 30 minutes to an hour, we are now doing it in essentially right at the source. So these components are only mixed together for less than a minute before they hit the source, and we're getting heavy chain and light chain reduction. So we're seeing the constituent parts of the antibody without having to do a kind of a benchtop reaction. And so, we wanted to combine this with ion mobility to do the CIU workflow for essentially for different types of reactions. One of the reactions that we did, moving away from small molecule reagents, but going to actually custom enzymes. So this is an enzyme from Genovis called Fabricator. If you're not familiar with it, this is kind of like an antibody cartoon structure here. Fabricator cleaves at a very specific spot at antibodies. With most antibodies, it has to have a sequence that it finds, and then it cleaves there. Essentially, you get 2 resulting fragments. You get this chain right here. It's called Fab prime 2. And then these 2 are the FC, or crystallizable. This whole thing is the crystallizable fragment. But when I say half FC, because now this is just essentially only non-covalent interactions, it's not uncommon for it to fall apart when you introduce some energy. So this is the resulting spectrum. Essentially what I'm doing is I'm infusing a mixture of these 2. Normally this reaction, like I said, this reaction will take 1 to 2 hours at 37 degrees C on the benchtop. But essentially we mix, draw it up in a syringe, and just start infusing it and run the collision-induced unfolding experiment. So we're stepping up that voltage. This is the resulting spectrum from one of the time frames. As you can see, the mass spectrum itself does not look very different. There's some differences in intensity, but the antibody that I stressed for 8 days at pH 3.7 versus the antibody that is in a ammonium acetate solution, largely the mass spectra look the same or very similar. But when we look at the different unfolding patterns, we can then start to see, so this is the Fab prime 2, that's this fragment that we're cutting off the antibody, and then we're essentially subjecting it to that, in this case, we subjected all of the antibody charge states and fragments to the energy, but I was just drawing on the 24 plus charge state of the Fab prime 2. And you can see we do get some interesting differences between the 2 different antibodies that were antibody fragments that were not stressed and stressed. And so especially at the beginning, we kind of have this 2 conformers here. Obviously, we have a dominant conformer and a less populous conformer there. But it tends to switch with the stress sample. And then going up here, we start to get these unfolding transitions at around 150 volts of energy. And then right around 200 volts of energy, we start to see that final transition as most of the population moves up into this most unfolded state. However, the stressed antibody sticks around, this conformation sticks around until about 350 volts. And then we see this this transition here at about 350 to that final state. Once again, drift time versus collision energy. Now, this isn't telling me what in the structure is changing, but it's definitely telling me there's a change in stability. So this could be used to go back for a test, to go back and then do a deeper dive if that was needed, kind of on what might be happening structurally, or what might be happening to the amino acid sequence to cause these structural variations. But this is kind of one example. We have some more enzymes that we're interested in that cleave at different portions or cut glycans off of antibodies. So this is kind of the gift that keeps giving, because there's always more reactions to look at. There's always more things to try.
Thank you guys so much. I was invited by Kasia from Bruker to kind of talk to you guys about how we're using trapped ion mobility spectrometry, mass spectrometry in the Wijayahena lab. So we're at NYU Langone here in New York City. And just to kind of give you guys some motivation why I'm so intrigued by ion mobility mass spectrometry is I deal primarily with proteins. So in actual cells, you have thousands of different proteins all together at once. And this becomes increasingly complex when you consider that some of these proteins have modifications, even more so when you have multiple modifications on a single protein. And it increases in complexity when you look at particular modifications, and they're also heterogeneous. So we really need a biophysical method to study and characterize the structure of proteins without disrupting their modifications in a high throughput and systematic error. Normally, NMR crystallography, kind of the gold standard for getting structure, is limited to like purely homogeneous samples. So I really think mass spec can handle this kind of heterogeneity problem. And just kind of highlight some of the keys. So just in general, ion mobility it identifies your protein by your mass with your mass spectrum, and then you could either identify structure or separate structure based on the ion's mobility. So here is just kind of like a very simplified workflow. If you have your LC retention time, if you look at a specific retention time and you look at your ion mobility spectrum, this could be to any instrument. What you'll see is that you have these different populations occupied. And what's really nice is if you have a case where you have an isobaric ion, an ion with the same M over Z, you could possibly mobility separate them. And we can leverage this ion mobility to basically take MSMS spectra of each of these populations, which I'll get into more. And specifically, we're using TIMS to do our ion mobility, and specifically, we're using the timsTOF HT. To just kind of give you guys an idea of what it looks like, I don't have an instrument schematic, so here's the actual instrument itself. This is the timsTOF. We have two different LCs that we're going to use. We have a NanoElute 2 and an EvoSep 1. For all the data that's I'm going to show here, we're only using the EvoSep 1, but still wanted to mention that they have other LCs that you could use. I wasn't sure the familiarity with TIMS, so just really briefly and quickly, I want to go through how the TIMS separation works. So in the most simple way, if we consider we have two ions here, one that's relatively large and one that's, say, compact. They'll enter the TIMS analyzer. So we are going from a higher pressure to a lower pressure. So we have that flowing buffer gas. And again, we're usually using nitrogen. And then counteracting this gas flow, we have an electric field that we're imposing. So what's going to happen is these ions, the larger ones, are going to move farther into the TIMS analyzer, and the smaller ones are going to be closer to the entrance. They're going to reach a point at which the force from the flowing buffer gas is going to equal the force from the electric field, and these ions are going to basically be parked. So hence the name trapped ion mobility spectrometry. That's where it comes from. Now we want to elute these ions from the TIMS analyzer. So what we're going to do is we're going to decrease the electric field at a specific rate. So what we do is we decrease the electric field. Larger ions, the frictional force is able to push them through the TIMS analyzer to the detector, while your other ions are still trapped in the TIMS. And you just continuously decrease this electric field and until all your ions elute. And this would be one single TIMS scan. And if you wanted to alter the mobility resolution here, we can't really extend our TIMS because it's a fixed cartridge. So the primary way that you would do this is to decrease your electric field at a slower rate. It was really nice that Thomas pointed out, because they're using a drift tube, they could get absolute mobilities. Here we have a dynamic electric field, so we have to use a calibration. So in TIMS, we're measuring an arrival time that we then calibrate to mobility or cross-section if you were interested in that. So we can leverage those ions leaving the TIMS analyzer. So you have two different MSMS techniques. You have data dependent. So if you did like a traditional, just pick your highest precursor and do MSMS, it's very inefficient. You're only isolating one ion at a time. But in the TIMS, what you're doing is you're going to do an MSMS as the ions elute from the TIMS analyzer so that you can get multiple MSMS spectra per TIMS scan. So it's much more efficient. You could also do this in a DIA manner. So instead of selecting precursors that are the most abundant, you could just set fixed mass windows to do DIA analysis. I'll only show a little bit of data here, but it still wanted to point it out. So I have a few topics. The first two being a little bit short, but I just wanted to kind of give you a few different flavors of how we're using it, kind of hiding the technical details because I didn't think they were too enjoyable, or too entertaining. But first, I just kind of want to give you guys a practical, practical use of the timsTOF and how we're using it in a proteomics lab. So we've had the timsTOF for a little over 3 years now. And in that meantime, we've thrown plenty of samples, cell lysates, CSF, plasma, purified proteins, toxins, thrown a lot of stuff at it. And over the course of 3 years, the QCs have been really good. We use HeLa digest to do our QCs, 200 nanograms. And over the course of 3 years, the mass spec's only good if you can continuously use it. And if it's down, that's not ideal. So we've been really happy with how well it's been performing over the course of quite a few years. And just in case you were curious, I wanted to give you some numbers in terms of protein and peptide identification. So we use a 3-organism mixture to kind of benchmark our instruments. And in this case, the human is always going to stay constant. And then we vary the E. coli and yeast concentrations. And just to give you some protein numbers, in the 200 nanograms total of protein, we're getting about 6,700 or 6,800 proteins and almost 63,000 peptides. This is doing a 30 sample per day LC separation, so 44 minutes. So you could go even longer here and you'll probably increase these. But what I really like is that we're still able to see proteins unique to E. coli and yeast at very low concentrations in the presence of high background. So here they would only be 10 nanograms and 100 nanogram background. So pretty happy with the sensitivity of this instrument and the overall performance in protein identifications and peptide identifications. Next, I kind of would switch and pivot away from proteomics and go into immunopeptidomics. And two of my coworkers, Iwana and Richland, both at NYU, kind of take these projects over. And just to kind of give some really surface level background here in the importance of immunopeptidomics. So it involves these HLA MHC peptides. These peptides are endogenous, so they're naturally occurring, and they appear on the cell surface. And this is basically what the T cell sees inside the cell and assess how healthy or diseased it is. So these, again, these are endogenous peptides. So the challenge really here is in the sample prep. You can't do a typical bottom-up workflow where you do reduce, alkylate, digestion, because then your tryptic peptides are going to interfere with your endogenous peptides. So you have to do a special sample prep here. And the problem is that it's very inefficient where you get massive sample loss and your MHC and HLA peptides are only a small fraction of that actual sample. So I mentioned endogenous peptides, and why that's important is because they don't always have a basic residue. Tryptic peptides always have a K or an R, so they usually have a charge state of 2 or 3, but these can have a charge state of 1, which poses a problem for MSMS. So in I'm in the TIMS, the way that you select the precursor ions is for MSMS is you kind of draw this polygon region. So you can not only select by M over Z, but you select by the mobility as well. And you probably saw in a lot of Thomas's diagrams, these kind of streaks across the ion mobility spectrum. And for peptides in this case, what these correspond to are the different charge states. So here you can see that the charge state 1 band for these immunopeptides is really prominent. Charge state 2, less so, and then not a ton of charge state 3 here because they're relatively small. They're only about 9 amino acids in this case. But what's really nice with the TIMS that we're able to do, since we can select based on mobility and M over Z, we can change our polygon region so that we can select charge state 1 ions with a mass above a certain criteria. We don't want to fragment these ions down here because they're likely not peptides. They're likely just interference. So we could actually gain some identifications by trying to sequence these charge state 1 ions. It's still very difficult because you typically only get 1 ion series, but it is still possible, and this is how we are doing our immunopeptidomics at the moment. So this is really kind of a nice way to do it. And just trying to summarize this workflow. So you have your sample prep. The end goal in this analysis is basically you're going to get a list of identified peptides that could be MHC candidates. You then do, in this case, we found about 1,400 total. You do a deconvolution to see what the common residues at specific positions here. So these are all NYMS. So the way that you would see this is that any amino acids on this side of the y-axis are upregulated. Down here means they're deregulated. In this case, this was an MHC class 1 antigen, so it has fixed anchors at positions 2 and F that are highly conserved, and we can see that there. You would then use these lists of peptides as possible candidates for immunotherapies. And I know the group here is using AlphaFold to kind of see which candidates that they can pick out of these lists, because only a few of these actually probably are the tumor peptide. So to kind of now pivot all the way back and talk about what I'm really excited about is and how we're using the TIMS in one application is combining affinity purification experiments with hydroxyradical protein footprinting. And this is going to be a collaboration with the University of Chicago, the Ruthenberg Lab. And to just kind of give you a general idea of how this footprint, like the principle of this footprinting is, say you have a protein and then a protein in a complex. This gray protein is the same in this case. What you're going to do is you're going to generate your hydroxy radicals, and then you're going to dose the protein structures with varying concentrations of hydroxy radicals. These are going to form oxidation modifications on amino acid side chains. And what you can kind of think about is you're painting the surface of the protein. And then you could think if you had a complex, you could then paint the protein in the complex. And basically, really simply, where you're looking for is where the paint disappears. So in this case, we're doing a LCMS, and you're basically going to quantify the oxidized peptide and quantify the wild type peptide. And you do this for several different doses I mentioned. So if the oxidation truly represents a structural feature, we would expect a dose response curve with the hydroxy radical concentration. So the result that we're looking for in these type of experiments are clear dose response curves, something like this, and there are some cases where you don't see as nice of a dose response, and in that case, like for this instance, you have a bit more noise. You can't certainly say that it is surface successful or buried. What's also really interesting is what you don't see in this plot, because if you don't see any oxidation, you can assume that this residue is buried in the overall fold of the structure. Oops. All right, and why would we want to use ion mobility? So in this case, ion mobility is not always the best thing for it, but what I really like about the timsTOF here is how fast we can acquire MSMS spectra. So in this example here, say you have this peptide here. You could have oxidation occur on many different residues, resulting in oxidized isoforms. And if your MS2 frequency isn't high enough, or if you have a high dynamic exclusion, you might not get proper sampling at the peak, because some of these oxidized isoforms elute very close to each other in the retention time, while others elute a bit farther. And basically what we need is a really fast mass spec to get many data points across the peak. And here I'm just showing kind of like a quality control, comparing the timsTOF number of MS2s at a fixed gradient length, and we're comparing it with some of our Orbitrap-based instruments that we have in lab. And you can see in 88 minutes, you take almost 140,000 MSMS, which is perfect for this type of experiment. Another thing I really like is if your source conditions are a little too hot, you could have in-source oxidation, which is an artifact. And the way that you would look at that is look for a peak in the oxidized peptide at the exact point where the wild type elutes. And what's really nice is that the unmodified peptide fits kind of right in between those two oxidized. So we don't think that the oxidation around this peak is coming from too hot of a source. And I mentioned this collaboration, so just kind of give you a biological background real quick, even though this is kind of more of an in-depth analytical technique. They're interested in this important 9 protein. This is a cargo protein. Basically finds cargo and transports it across this nuclear envelope into the nucleus. And this particular cargo is really interesting because it's critical to cell homeostasis. In mice with depletion, I think it was lethal in 10 days. So it's very important for cell homeostasis, but they don't necessarily know all the cargo that it's bringing it through. And they're also interested in where this cargo is binding, because this IPO9 doesn't have a classical binding motif like some of the other importants do. So they did a affinity purification with flag grown IPO9 in HEC239 cells. And then we also have controls that lack the flag IPO9. And the Ruthenberg lab was responsible for preparing these samples. Next, to kind of go into the workflow, so we have these proteins attached to a bead. So we have our magnetic beads, the IPO9 and its cargo all attached to the beads. What we're going to do is we're going to footprint these proteins while they're still on the beads. And what's unique about our lab and the way that we do footprinting is that we do it with pipette and benchtop chemistry. So you don't need any specialized lasers. Anyone with common lab equipment can perform this footprinting reaction. And I mentioned before, we do varying doses of hydroxy radical, and we typically do at least 3 replicates. I sometimes like to do 5, but you need at least 3 replicates to get good statistics. Then you would just proceed with your quenching and cleanup, and we're going to use the EvoSep in this case. In case anyone isn't familiar with EvoSep technology, it's an LC that does the C18 cleanup online. So in this case, it just kind of speeds up the workflow a little bit. It prevents cleaning, drying down, reconstitution, and stuff like that. And we're using a bionic to do our analysis. And again, we're basically going to look at the oxidation area over the total area of the oxidized and unoxidized peptide. So the ending result here is I'm showing a bunch of dose response for particular peptides in the IPO9 protein. What's really nice is that we see several clear dose response curves, so which tells us that the integrity of the structure wasn't completely destroyed when we would footprint. If the structure was really kind of disrupted prior to footprinting, we would expect kind of dose responses like this here, something not very clear. This is very expected when you have a really long peptide, because usually your MSMS sequence coverage there isn't the best. But we do see a plenty of strong dose response. And then again, what's always interesting isn't what's always there. We do find several peptides that are abundant, showing the average intensities here, but are not found to be oxidized at all. So these peptides could be in the binding interface between IPO9 and the cargo. So it's kind of indirect information, like what you don't see is still pretty interesting. And next, so I could take that, you take that same data and just kind of transform it a little bit. So here is the same data that you saw before, but now the oxidation response is plotted along the amino acid sequence. Down here, these black bars represent the areas that we just don't detect. We can't comment on those areas if we don't detect them, but we do see several regions that aren't oxidized and several regions that are oxidized. And kind of very qualitative kind of viewpoint, I can take these oxidation percentages, superimpose them on a 3-dimensional structure, and kind of get an idea of, are we in the right ballpark? Is this surface accessible? Now, this isn't quantitative at all. This is purely qualitative, just kind of looking at it. And I do want to mention that the oxidation typically occurs on just a couple of residues. And for visualization here, I'm stretching the oxidation across the whole peptide. Realistically, it's not the entire peptide that's oxidized at every residue. It's only typically a few. So, and the color coding here is just to show the white see-through regions are the unoxidized, and then the red regions are oxidized. Next, I mentioned that we wanted to also identify cargo, but what's really nice is with this experiment is you could just take the same samples and run your traditional DDA experiment and get the PSMs, the peptide sequence matches for each protein out of each of these samples. So what we do for our analysis and binding partners is we create these one-sided volcano plots. So on your y-axis here, you have a SYN-R score. This is just our version of Saint Express. So this is a statistic. And the way that you would interpret it is that a value of 1 would mean that it is a highly confident binding partner. And you kind of see these plots kind of have different levels here. What this corresponds to are binding partners found in 1 replicate, 2 replicates, and then up here would be binding partners found in all 3 replicates. And then on the x-axis, you have your fold change. So I just selected the bait, so our IPO9, and a few of the binding partners here. And you can see in our controls or empty vectors here that we don't really have these protein, and it is relatively abundant in the pull down experiments, suggesting that this is a real cargo. And then to kind of piggyback back to the footprinting, what we could do is say you are interested in a particular cargo. You can then look at the dose response here. And we can see even for the cargo, we're able to see several dose response curves for several peptide regions. But also what's really interesting here, again, is what's not here. I'm not showing the list of peptides because it's not nice for a presentation, just a table of peptides. But if you don't see the peptide oxidized on the cargo, that could be the binding interface. But you can't rule out that it's also in, say, a hydrophobic core of a folded protein. So just to show, you could do this. I'm doing this for 1. You could do this for any of the proteins that you're interested in. And you can only really kind of do this for proteins that are somewhat abundant. So if you only have 10 to 20 PSMs identified, it's hard to get a nice dose response curve, but it's still promising to us that you can still see some dose response, although some of it is a little bit noisy, but still was really surprising to us. And to our knowledge, this is the first time that footprinting on beads has been tried on the benchtop without the use of like lasers or anything like that. So just to kind of wrap things up real quick, I'm sorry if I went over. The general idea is that you can paint these surface or protein bound to beads and its cargo and look at its binding partners, and then thus look at the oxidation dose response for each of these binding partners. And just want to briefly wrap up and mention kind of this is ongoing, but I'm trying to improve it, and the main source of improvement really has to do with the footprinting chemistry itself. So it's done on the benchtop, so it's subject to human error and pipetting error. So as good as I think I am at pipetting, I'm still going to make mistakes. It's just kind of what happens. So to solve this, the idea that we have is to replace the human user and stick in a robot. So we got an OpenTrons Flex, which is a robot that you supply a Python script, and it will perform your benchtop chemistry for you. So it kind of has a deck set up like this. And the general idea that we eventually want to get to is what you're going to do is you're just going to put your protein here, add your reagents, click start. This protocol can run all the way up into the EvoTep loading, so that when the protocol is over, you could just pick up your tips, put it on your instrument, and click start again. And this, if just a shameless plug, hopefully I'll be presenting this at ASMS 2026. If you're there, come by. And then just lastly, want to acknowledge all the people that worked on the project again, the Wijayahena lab, especially Ioana for the immunopeptide stuff, and Richland, who's also working in our lab. Collaborators at the University of Chicago, Ruthenberg lab, especially Amanda. She does a lot of that sample prep that you saw with the affinity purifications. Kasia from Bruker for inviting me, and of course you guys for allowing me to come speak to you guys today. Thank you so much.
I'm from University at Buffalo, and I got invitation from Waters, Noel Elliott. So I'm not going to talk about protein. I'm going to talk about PFAS. Hopefully, I will give you a brief introduction to PFAS, and let's move on to the isomers. So my topic is PFAS isomers matters, distribution pattern of linear and branch PFOS and PFOA in consumed fish revealed by cyclic ion mobility separation. So this paper, actually, this is the title of the paper that I published last year. So it's recently came out. Can check it out too. And I work under Dr. Diana Aga. So as I said, this is just a brief introduction because I think most of the people have heard it. But if somebody is new to PFAS, PFAS are the shorter term for per and polyfluoroalkyl substances, and they also have the common name called forever chemicals because they have this carbon-fluorine bond, which harder to destroy and harder to degrade in the environment. So if we think about where to find this, it's in everywhere. If you eat pizza, you get it in, and if you have a raincoat, waterproof, then you have PFOS. So it's basically everywhere. And when I say PFOS and PFOA, PFOS and PFOA, if somebody's new, you can see here the structures here. So I'm more towards focusing on these two compounds in this presentation, but I will kind of show what's in fish briefly. So why do we care? Of course, all these compounds at the end ends up in our body. So there are a lot of health effects. Am I moving on? Okay. So where to find PFOS? It's everywhere, as I said. So this is just a map that showing in US how contaminated our water is. So it's basically when you drink, when you eat, it goes into your body. So this is the bottom. You can see a plot where given by, this is by CDC, where they're showing nearly all the people in US have PFAS in their blood. And PFOS, PFOA, PFHXS, and PFNA are the common. So it's going down because of the phasing out, because now there are regulations on this. But still there, it's persistent, as I said. So then we were like, okay, our water has it. So what would be the dietary side that contaminates us? So the one of the major matrix came to our mind is fish. Fish, because that's one of major nutrition sources that people including in their diet to get all the nutritions, and every year by year the consumption is going up. So we were like, let's see what's in fish, right? So we went to the market. This is just a rough, a brief diagram what we did with the fish. So we basically went to the market, bought it, and then crushed it, followed a modified EPA 1633 method. When I said modified, is one of the major modifications we did was we used this new wax, GCB on the top, and wax cartridge. And we had some alterations throughout the method, but then at the end, the fish extracts were analyzed using Waters Equity Ultra Performance Liquid Chromatograph with a select series cyclic ion mobility quadruple time of mass spectrometer. So QTOF, UPLC, CIMS, QTOF, MS. So again, we used Waters application note for our LC method. I'm not going through this in detail because you can find it in Waters application note. Basically, we used that with a little bit of a modification because we used a different C18 column than in the application note. So after we ran our fish samples in the IMS, you can get the mobilogram, where you can see how the ions have traveled throughout the drift tube in the CIMS. And you can clearly see this is a trend that already been identified, where you can see the per and polyfluorinated organic compounds in the bottom, where this includes all the PFAS say PFAS in the sense all the fluorinated compounds and all the other compounds on the top. So this is also opens the doors to, like, if you are like targeting compounds, of course, those compounds should be in this line, as well as, although I'm not going to talk about here, non-target analysis, because you can like identify what each these compounds are. And then just roughly touching on through this, we found like 13 out of 1633A EPA method PFAS. Just wanted to say every fish had PFAS. So you basically, when you eat fish, you are taking PFAS into your body. Tilapia had the lowest, rainbow trout had the highest, but I want you to focus that all these fish has PFOA and PFOS, and 6,2-FTS was detected, but mainly the legacy PFASs were there, because in the published world also they say like these are the two that mainly detects in everywhere in every matrix. So then, one of the other known things is they have isomers. It's new now to the field, but it is like now an emerging field in research where these PFAS and PFOA known to have like isomers because of their production method. So they produce through electrochemical fluorination, which leads to linear and branch, where linear is the highest, of course, but branch as side products. But people did not care in the past, they only care about the property of the isomers. And then the method developed accordingly to identify linear PFAS and PFOA, actually PFAS commonly. Then later, they also seeing like isomers also build up in our bodies, bioaccumulating, persistent. So that's where this another door for PFAS experimentation opened up. So you can see I gave most of the structures that is known so far for each PFOS and PFOA isomers, because I want to let you know PFAS is also like a broad class that consists of over 14,000 compounds, and imagine isomers in each compound because of the producing method. So PFAS, the other thing I also want to emphasize is there are not much studies done on health effects, like how it differentiates from linear PFAS to like branch PFAS, but there are few has done, and also it's an emerging field, as I said, and people have seen, researchers have seen linear PFAS and branch together have a wider, like a broader effects on proteomics. I'm just giving this sentence from one publication, but there have been a lot of studies have been showing linear to branch having diverse effects in the health. So moving forward, I know it's like everybody has known how the NYMS separates the ions, but I like to show this because we have the cyclic ion mobility separation where the ions goes through a cycle, and then we can manipulate the number of passes it goes through the water's CIMS, and we can separate the PFAS isomers. So this is one of the important character we want, because PFAS isomers have a very like close CCS values. So you want like higher passes, like more separation ability to get the PFAS isomers separated. So the process that I showed in earlier slide happens in here. So you basically inject your samples and goes through the quadruples, like you select your parent mass, and then you passes through this cyclic IMS as much as you want to get it separated. So like that, again, I'm showing the parameters we actually developed for the PFAS analysis. And this is also a recent publication from our group, which also have how we optimized all these conditions, how these conditions affected our PFAS isomer analysis. And then moving forward, it was very excited and interesting to see. I know having fish, like PFAS, like fish having PFAS is a bad for us, but seeing this is very interesting, because if you inject a standard of PFAS, we had to optimize 6 passes in the Cyclic IMS. So it goes 6 passes, and then we were able to separate linear, and all these are mono. So P1, P2, P6 are like the position where that methyl group is. And in the bottom, you can see the dye. And in benthic fish, I will later show who are the benthic fish and pelagic fish. But in benthic fish, we were able to see like higher amount of PFAS. And in pelagic, it's very less. And when moving on to PFOA, we had to do 3 passes, because when it comes to PFOA, it has a carboxylic head group, which immediately fragmented from the compound, and it just gave us the fluorinated tight chain length. So it needed like 3 passes. And I know here we were able to separate mono and did not see any dye here. And in compared to benthic and pelagic, P4 isomer profile, they doesn't have that much of a change like this due to its solubility in water, because they are in fish, so doesn't do a good partition between linear and benthic. In the other hand, there are research studies have shown linear and PFAS basically tend to accumulate in the sediments, which benthic fish feeding on. So we saw that difference where pelagic has lesser PFAS. Tilapia is one of the pelagic fish, and haddock has lot, while PFOA doesn't have significant difference. So with that, the other thing we wanted to see is, are we quantifying these PFAS correctly? Because in EPA 1663A, they say like report the total PFAS or total PFOA like that. But are we like then correctly quantifying? Are we overestimating or are we underestimating? So we wanted to see how each of these isomers ionized in the mass spec. So you can clearly see linear one had the lowest relative ionization energy efficiency than the others. So we are actually kind of like overestimating if you're using the linear PFOS calibration curve or like isotope dilution to do the quantification of isomers. So this work was able to do with the CIMS because we had individual isomers from Wellington, and we created calibration curves. And for the first time, we showed like, no, it is better to have own isomer standards when you quantifying the isomer profiles in biological or like any matrices. So with moving on to our fish analysis, so we saw PFOS isomers in fish, all mono substituted branch PFOS were observed in benthic fish. As I said, PFOS like to attach and bioaccumulate in sediment. So then benthic fish goes feeding on it. So this kind of approves our hypothesis, and also what literature has found that they have more exposure to PFOS. And the benthic are the ones that are highlighted here, and these are the pelagic. And for the reference, I have a PFOS standard, so you can clearly see the isomer profile is totally different, and also isomer profiles are totally different from benthic to pelagic. But overall, if we consider the PFOS all concentrations, isomers plus linear everything, benthic had higher PFOS, again confirming what literature has found. And when it comes to PFOA isomers, as I said, because of its pKa and the higher water solubility because of the carboxylic acid, we did not see a significant difference between benthic and pelagic. And it's also kind of around the what you say in PFOA standard. So with that, it was like for the first time we were able to like publish a paper with all the in-depth analysis from like each individual isomers from legacy PFAS, which is PFOA and PFOS, and it is already published. So as a conclusion, with the water CIMS, it's like gives us more opportunities to separate isomers. So in this scenario, we had like different passes for both the PFOS and PFOA, but we were able to get good results and separate isomers. And fish, yeah, it's sad to say, but they have PFAS. So detected linear and mono, and we had higher like PFAS in benthic fish than pelagic. So if you're eating fish, go more towards pelagic fish types. And then we had a different isomer profile for PFOS in benthic versus pelagic, but not with PFOA because they kind of did not have significant difference. With that, I would like to acknowledge my group members and my advisor, Dr. Dayanaga, and the funding sources and IMS in our renew. So thank you all.